Hello and welcome back. This lecture is the start of Chapter 10, From DNA to Protein, Gene Expression. Sometimes students struggle with this chapter because we will be looking at how protein is produced from DNA. This can be a very visual chapter, so I recommend that you read the chapter and watch the videos that I post on Canvas to help understand the material. So let's hop right in and get to it. This chapter has an opening question that we will be able to answer by the end of next lecture. The question is, how do antibiotics target bacterial protein synthesis? We will find out by the end of next lecture, but first, a little bit of background starts the chapter. It is important to note that not all genes code for polypeptides. If you remember a while back, polypeptides are chains of amino acids that make up proteins. Some genes are transcribed to RNA, but not translated to polypeptides. These RNA have other functions. Molecular biology is the study of nucleic acids and proteins, which often focuses on gene expression. This chapter will just touch upon gene expression, but molecular biology is a vast and growing field. Genes are expressed in two steps. Step 1 is transcription. This is where information from a DNA sequence is copied to a complementary RNA sequence. Step 2 is translation. This is where the RNA sequence from transcription is converted to an amino acid sequence. The diagram on the bottom of the slide is a handy guide for this chapter. DNA is transcribed into RNA. RNA is translated into polypeptides. We'll get more in depth with these processes in a little bit. There are three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA, or referred to as mRNA, is a complementary RNA strand produced from a sequence of DNA. The mRNA moves to a ribosome to be translated. The nucleotide sequence of the mRNA determines the sequence of amino acids in the polypeptide chain. You can see an mRNA on the top right as the yellow strand. It looks like half a strand of DNA with uracil instead of thymine. Ribosomal RNA are often referred to as rRNA, along with some proteins make up ribosomes. rRNA catalyzes peptide bond formation. You can see a picture of rRNA on the top left. Transfer RNA, or often referred to as tRNA, binds to specific amino acids into a specific sequence of mRNA by base pairing. tRNA recognizes which amino acids should be added next in the growing polypeptide chain. It carries amino acids to the ribosomes. You can see a picture of tRNA on the top right. Figure 10.3 shows a diagram of DNA being transcribed to messenger RNA, and then mRNA being translated to produce a polypeptide chain. In eukaryotes, transcription occurs in the cell's nucleus, and translation happens in the cytoplasm. Here's a picture of the three types of RNA that we'll be talking about in this chapter. mRNA is on the left, with rRNA in the middle, and tRNA is on the right. Here's our first progress check for the chapter. Do your best to answer these questions. Once you're ready, continue on with the lecture. In order for transcription to occur, it requires several things. It needs a DNA template for base pairing, and 4 nucleoside triphosphates that would become adenine, thymine, cytosine, and uracil. And it needs an RNA polymerase enzyme. RNA polymerase synthesizes RNA from the DNA template. RNA polymerase is processive, meaning that a single enzyme results in hundreds of RNA bases being catalyzed together. Unlike DNA polymerase, RNA polymerase does not require an RNA primer. Here's a 3D model of RNA polymerase. You can see DNA in the middle is represented by the blue structure, with the green RNA being produced from the DNA template. Transcription occurs in 3 steps: initiation, elongation, termination. We will go over these steps in the next few slides. Initiation is the first step of transcription. Initiation requires a promoter, which is a control sequence of DNA that tells RNA polymerase where to start transcription, which strand to transcribe. Each promoter has a transcription initiation site where the RNA polymerase will bind. Other proteins will bind up from where RNA polymerase binds to help determine which genes are expressed. Figure 10.5 illustrates RNA polymerase binding to a DNA strand. Pink areas in the DNA strand are the promoter region, guiding the RNA polymerase where to bind. During the second step of transcription, elongation, RNA polymerase unwinds DNA and adds nucleotides to the 3' end of the new strand. The RNA transcript is anti-parallel to the DNA template strand. Uracil instead of thymine in the RNA molecule is paired with adenine in the DNA molecule. RNA polymerase can proofread and recognize errors. It allows more mistakes than DNA polymerase does. Final step of transcription is termination. Termination is specified by specific DNA sequence. Eukaryotes use different types of proteins in recognizing the termination sequence and separating the newly formed RNA from the DNA template. Figure 10.5 here is showing elongation and termination. Elongation is the addition of RNA nucleotides to a growing RNA strand, while termination is the removal of the RNA from the DNA template strand. Before this RNA strand can be transcribed, a few things need to happen. But first, let's talk about coding regions. Coding regions are sequences of DNA that are expressed as proteins. In prokaryotes, most of the DNA is made up of coding regions. In eukaryotes, each gene has its own promoter. Most genes have non-coding sequences called introns. The transcribed regions are called exons. Introns and exons both appear in the RNA transcript. The RNA produced in transcription will become messenger RNA. Introns are removed from the final mRNA. Table 10.1 illustrates the difference between prokaryotes and eukaryotes gene expression. We will mostly focus on eukaryotic gene expression, but it's good to note the difference between the two. Figure 10.6 shows a DNA molecule with a gene. This DNA is transcribed into pre-messenger RNA, has both introns and exons embedded in it. Introns are removed from the pre-mRNA to produce mRNA. Introns need to be removed from the mRNA because they interrupt the DNA sequence of a gene. Most eukaryotic genes contain introns. pre-mRNA is modified by RNA splicing, which removes introns and splices the exons together. It does this by using sRNPs, small nuclear ribonucleoprotein particles that bind to exons and introns. snRNPs bind to consensus sequences, which are short sequences of RNA. Other proteins accumulate to form a large RNA protein complex called a spliceosome. The spliceosome cuts the pre-mRNA, releasing the introns, and joins the exons together to produce mature mRNA. Here in figure 10.9, we see two snRNPs attaching to a pre-mRNA molecule. The snRNPs form a spliceosome, removing the introns and bringing the exons together to form mature mRNA. The pre-mRNA is further modified at both ends. A 5' cap of guanine is added to the 5' end. Its purpose is to facilitate binding to ribosomes. A poly-A tail is added to the 3' end. This assists in export from the nucleus and contributes to the stability of the mRNA. And there you have it. That is how mRNA is transcribed from DNA. Here's our final progress check for this lecture. Take your time to answer these questions. Please watch the videos of transcription on Canvas as well. As always, let me know if you have any questions on anything. Thank you for listening and have a nice rest of your day.